Here, Mark and everybody. I see already a few familiar faces out in the audience, and I appreciate you coming to see me. And thanks. I don't know where the Zoom people are, but hi to you as well. So I wanted to talk to you in maybe a bit of a mishmashy thing. Hopefully, it'll make sense. Is much about kind of what I think some of the cool things that we're doing, but also there's a subtext of where I think various aspects of the field could be going. And what do we mean by the field? I mean, actually, life sciences per se. My background is in evolutionary genetics. I was an evolutionary quantitative geneticist, did a lot of population genetics, always tracked whatever the newest developments are, do a lot of population genomics now. But I also, about 15 years ago, started working more and more in aging, and my lab is now, thanks to generous funding from the NIH, we do a lot of aging research, and so I'll be talking a little bit about the mix of those things. Before I start, I want to thank my lab. So they surprised me. They went to JCPenney and did this awkward lab photos thing, which I guess is an internet meme, and so they put this together. And so now this is now our official lab photo. I won't point it out. Many different people contribute many different things, and I'll highlight a few folks as we go, but not everybody. So this is a kind of traditional way to start an ecology and evolution talk with some sort of thing. Frequently a biodiversity thing with all different kinds of organisms. I thought, well, I'll just show the coral reef, which is another archetype of what biodiversity is. And really biology and why I became a biologist is, how did this happen? Like what, and there's many different aspects to the how. One is the historical thing about how these organisms came to be, but then also, how does this fish develop? And then the thing that's always fascinated me is, as we look across this vast display of things, how are all these individuals the same or different? What creates coherence and discoherence in all of that pattern. So when we look within a population, which has been most of my history, then we want to know kind of what makes everybody different. And of course, in the medical sciences, this is becoming more and more of an issue. Instead of treating kind of the generic person, we're very interested in personalized medicine. So there's, from an evolutionary point of view, historically, this has been addressed by using what I would call cross-sectional analysis, where you just ask, how does a population change from one generation to the next? And this is, I think, the key equation from evolutionary genetics, which is basically the change in allele frequency. It's just it's a counting exercise. How much more fit is one allele than another one? And then it increases or decreases in frequency depending on that thing. And so there's no sense of history in that. It's just, you kind of just keeps restarting and tracing that. And to some degree, that was a mathematical convenience, but also because we were trying to tie this to genetics, which was a transmission-oriented activity. Now, somewhat ironically, Charles Darwin is, of course, famous for coming up with the idea of natural selection, upon which all of these ideas are based, only had one figure in The Origin of Species, and that's a phylogeny. And it's as important as natural selection is an idea, and that clearly Wallace was simultaneously coming out with it. I think you probably all know famously, is really the idea of Darwin that I like the best is descent with modification, because that captures all of evolution. And then natural selection is one way that things become modified. There are others. And so it's really the fundamental idea of Darwin is how all of these draw together and how there's a unity of life. And that's a historical statement that is driven by other causal elements that I think are important. And so this is a recent paper looking using whole genomes to create a phylogeny of flowering plants. And again, it begs the question, how do all of these things, how are they related? How do they become different from one another? And some people are just like really into the history, like what is this connection? When did that node happen? But also many people are interested in what were the evolutionary mechanisms that drove that. For flowering plants, we've frequently think about pollinator dynamics and things that were going on with that. So to me, there's this essential tension in any kind of explanation about why things happen. And it's this dynamical explanation versus historical. And I think that we have never quite got that balance correct. And that's partially because the approaches and the information have never really lent themselves to do that. And I think we're at the cusp of that fundamentally changing. As you can see, just in that flower example, there are people who are studying selection on flower color and pollinator dynamics at the genetic level. At the same time, we're looking at the genes that create relationships among flowers. So from a dynamical point of view, we have things like population quantitative genetics that use these historical cross-sectional approaches, generation by generation. We have inferred history, which are obviously the domain of phylogenetics. Population genetics has, over the last 40 years, moved into that using coalescent theory, but that tends to have more of a longitudinal idea. And to me, that actually is the fundamental way of thinking about evolutionary change, is again, descent with modification. But there's a whole other parts of biology that are not evolutionary, of course. And we frequently can describe those in terms of natural history, like what is actually going on with organisms. But that also encompasses what I would call functional biology. And I would actually put molecular biology as a component of this. In many respects, a lot of molecular biology is the natural history of the gene or a protein. Is how does it work? What does it do? What is its context? And of course, we use very strong experimental frameworks for getting at those questions. And so the final thing that I'm going to emphasize, and I won't be able to really explicate this as much as I would like, is I think that the next generation of biology is actually the need to create new biology. This intermingling of dynamics and history and function are so hard to separate that in order to both understand some of these fundamental ideas and then to address things like some of the challenges that we'll talk about with it, for instance, in aging, we need to create new biology in order to understand the biology that we have. And so that's what my lab is really about now. So as I said, about 15 years ago, we started doing more and more work on aging, and we've really expanded on that. So I just want to give just a brief introduction to aging as a concept. This is our friend Charles Darwin. Turned out that your beard just keeps growing as you get old, and so that's a fundamental feature. So the basic kind of atom of this aging research is a survivorship curve, where we look at the probability of survival over time. So imagine you start with a cohort, everybody's alive when they're born, and then it changes over time. So if we look at a human population, we want to see what that is. So the really important idea is that if there was a constant rate of mortality at every instance, then you would get this negative exponential relationship. And so that if nothing changed over your whole lifetime, and you can think of this as the bus theory of aging, if the only way you're going to die is you randomly hit by a bus, then you would have some survivorship curve that would look like this. This is distinctly not what human survivorship curves look like. Matter of fact, most organisms don't look like this. Some do, but not most. So in the United States and in most developed countries, the survivorship curve looks something like this. And there are sex differences. So most of the time you live with a very low rate of dying for a long time, and then something changes. And in humans, it's around 62 years old. And then something else happens. And so aging is not just getting old, and it's not just a chronological event. There's a fundamental shift in the probability of dying at a certain age, and that starts to accelerate. Okay. So if we took a 60-year-old man and then looked at took that rate of mortality at 6 years old and just stretched it out, it would follow this black line. So if mortality did not accelerate past 6 years old, humans would actually live to be, or these men would live to be about 250 years old. Okay. So if you could just stop aging at age 60, you live to be 250 years old. So that's what makes this field enticing to some people. It's like, well, that seems possible. So what happens, do you think, if we stopped the rate of aging at 30 years old? So you'd live to be 1,800 years old. So when there's a lot of money, billions of dollars now going into longevity research now, and that's because there are humans on this planet right now who, if we could somehow capture their rate of aging in a bottle and then resell it, they would live to be 1,800 years old. So the question is that biologically possible or is it realistic? Why doesn't that happen? And that's really the big question of the field. So we call that accelerated in the rate of aging as senescence. So this has become quite the thing to do over the last 30 or 40 years. And there's many different causes of aging. I'm going to highlight just a couple of things from phenotypes that we're looking at. Some of them are related to oxidative stress, which can be related to mitochondrial dysfunction. We're also doing some things with genomic instability, but mostly what we tend to look at, because as you'll see in a second, we work with these nematodes, are things that occur from the cell membrane in, because that is universally shared across all eukaryotes. The way that the cells function is pretty much the same. Humans have a lot of other things going on with them that worms don't have, like hormones and immune systems and all this other stuff that are really important components of aging as well. So we work with this model nematode, C. elegans. I actually did, when I was doing my graduate work with Steve Arnold, I worked on frogs. I just decided that frogs were a crappy genetic organism. And so when I started my own lab, I wanted to be able to work on something where there was a lot more genetics. And at the time, almost no one was doing anything in evolutionary genetics with C. elegans. And so I thought it was a good potential model. Part of the reason why people study aging in C. elegans is this several discoveries that were made in the late 80s and early 90s. And it's basically this. So this is what a survivorship curve for worms looks like. Notice that this is measured in days, not years. That's also a convenient thing about worms, is you can actually study longevity in a reasonable amount of time. You don't need to have several human lifetimes just to study one cohort. So Cynthia Kenyon noticed this mutation DAF2, which is actually turns out to be an insulin receptor, makes the worms live about twice as long. And so this was a mutation that, rather than killing things and creating dysfunction, there was lots of mutations that are known to do that. It's a mutation that has a positive effect. And so this basically created this entire gold rush approach over the last few decades into understand the genetics of aging. And so that was an important observation. And because it turned out when this was cloned, it's an insulin receptor, turns out that this gene and the essential function of this is conserved across all animals. And so including, you can study it in flies or mice or humans or monkeys. And so that this seems to be a conserved mechanism for extending life. So part of the reason why we got involved in aging is because we were already studying natural variation in nematodes. And so kind of on a flyer, I submitted an R03, which is the world's smallest grant you can submit to the NIH. It's a $100,000 grant. And I said, hey, let's look at natural variation in longevity. And they said, okay, that sounds good. So we did that. And then that started us on this cascade of using that as an approach to understand the biology of aging, using this really important model system for aging research. So I'm just going to tell you, I think, 3 vignettes here of research that we have going on in the lab that are trying to get at some combination of understanding aging as a complex trait, understand it a little bit from an evolutionary point of view, and then also point the way towards the future ways that we're going to be looking at this. So we actually started working on a variety of stress responses before we even started working on aging. So this is resistance to paraquat. And so these are two strains, and I won't go into all the background of these. N2 is the standard lab strain. My16 is the another natural isolate. It actually is a European isolate. And so these have both been put on 0.4 millimolar paraquat, and it's kind of hard to see. I'm not sure if this is going to, nope, that's what I was afraid of. Where is the, it's, oh, the middle thing, of course. Okay, if you look at the worms grow up and eat this bacterial lawn here. If you look at this lawn, you'll see that it's kind of clear. There's just the worms. If you look over here, all these little dots here are eggs, okay? So My16 is able to basically reproduce on paraquat, and N2 can't. So if you measure that as an average fecundity measure over a short amount of time with a large population of worms, you see that My16 is highly resistant to paraquat and N2 is not at all. So we decided to understand what the genetic basis of that was. And so we've done a lot of different things with worms. For QTL, what are called quantitative trait locus mapping. We've used recombinant inbred lines, all sorts of stuff, but we wanted to start using this, what's called bulk segregate analysis, where you cross the different strains and then select for extreme individuals. And so that's something that's done a lot in yeast, for instance, and some people have done it in worms as well. So we decided to put our own spin on this by doing this over multiple generations. So we call this evolutionary segregate analysis. So we do the crosses, do a generation, and then keep selecting on the trait on the extremes so that we can create a stronger signal when we look at the genome sequencing of where the differences between the strains are. The other important thing about this, as you'll see in a second, is that there are other things that change between the strains besides the thing that you're selecting on. So in particular for worms, for instance, they tend to seem to have a lot of interactions just in their genetic background. So different parts of the genome just go up or selected for just who knows why, lab adaptation, all sorts of things are going on. And so in order to get the signal out that you want, you really have to have controls, and you need to look for it in detail. So what we did is look across the whole genome. Worms have 6 chromosomes. Just look over, well, basically, this is the change. Well, these are the significance values where we measure the change in allele frequency over 1, 2, 3, 4, 5 rounds of selection. And you'll see that there's different peaks and things going on. One of the features of worms is they have very low rates of recombination in the centers of their chromosomes and high rates at the edges. And so you always get lots more signal at the edges. So that's an interesting bias that you need to be aware of. And so when we did this over time, we can do it just by looking at the lines that were under selection, which is frequently a way that people have done these kinds of experiments. And we get a very strong signal on chromosome 5. But we can also then control for the background changes that we see, and we still get that signal, but it helps to contain the other things that are going on. So zooming in on chromosome 5, the black parts are from N2, and the other parts are from My16. And what you can see is that there's kind of a mishmash of things going on. But let's look at this end here, with it is almost all black, just in the controls. And you'll see that this, now that here are allele frequencies, that there's strong selection for the N2 chromosome in that part, just regardless, right? So it's really hard to pull out the signal from that. And so it turns out to be this section with the red bar where we see that each generation, so here's the ancestral values, which is still biased because I didn't go into it, but these lines were created by 20 generations of crosses to create recombinant populations. So this entire chromosome is really favoring the other genotype, the N2 genotype. And so by selecting on the paraquat resistant, we're actually pulling out the section of the chromosome that's responding. So you see this is getting oranger and oranger over time until after 5 generations, it's really biased towards that. So that allows us to focus in on this part of the chromosome. But even then, it's pretty big. It's many megabases. And so what we did is go through and kind of look at the fine mapping here. And then John Willis, the senior scientist in my lab who worked on this, kind of looked through, and we looked for likely candidates and so on and so forth. And we identified one. It's in a gene called GAD3. We'll talk about its function in a second. But basically, it had a couple of coding changes. And so we did what one can do now and was impossible to do even a decade ago, is we just use CRISPR, and then we just flip the alleles back in the different parental populations so that we could actually directly test whether or not these things were causal. And it turned out that it worked. Again, to me, when I was growing up as a little kid, this was like the holy grail to be able to do this kind of experiment. So we're really happy that we could do it. Okay, so when we do that, there's 2 directions we can go. It turns out there's actually 2 different amino acid changes, so we can substitute them each in. And when we substitute the My16 into the N2 background, just one of the changes, it definitely increases resistance. And then when we put them both in, they both go. So there's 2 different changes in the same protein that both seem to be important. And then when we step back the other way, we start with the resistant line, and then we move one, and then the other, we're able to drop it back down. We're still not getting quite down to the other parent, and that's because there's no doubt there's other loci with other effects that in that background that matter. So it turns out that GAD3 is in this pathway that's involved in oxygen production. And so what we think is happening is that there's a leaky Ross effect in the cell with one of the allelic backgrounds. And so that the cells are kind of pre-primed to be subject to this oxidative stress, which is generated by the paraquat. So it's what is sometimes called a hermetic effect. And so this is, to me, it really highlights how complex traits can be. So you have a leaky protein that is basically turning on oxidative stress response genes due to its actual physiological effect, which you wouldn't see and we're looking for now. And then it's the thing that is then making the whole organism resistant to oxidative stress challenges later on. So we looked at these 2 amino acids across the distribution of C. elegans around the world. And what we find is that they tend to be found together in different clades. So there's a clade that is mostly a combination that's My16-like, and there's a clade that's N2-like. And our My16 is actually kind of this intermediate clade, but it has both changes. So for the most part, these 2 amino acids tend to go together. So you're either one type or you're the other type, which is known as epistasis, which is a whole nother part of my life that I've worked on historically. So we could actually look at this GAD3 thing, which is an aldehyde oxidase, and we can model these changes onto the to the human homolog of this. And no great biochemical insights emerged, unfortunately. I was really hoping this would be like right fitting around something. So the 2 changes are on these different sides of this enzyme, and it's not totally clear, if it's doing some conformational change. It must be doing something that affects the overall activity or in interaction with some sort of cofactor. So we don't totally know what's going on. But the lesson here is that we can start from some gross phenotypic difference between these things and then pull it all the way through genetic mapping to doing genetics with the substitution to looking at the biochemistry of what these changes actually do. And so what we're doing now is actually trying to measure the actual oxygen levels in the ROS production within these strains and see if we can nail it down to test the hypothesis that we have that there's sort of a hermetic effect that's going on for these things. So that's the last little bit before we submit this for publication. Okay, so we have these different strains. It turns out these strains also live different lives. Whether or not these things are different is unclear. When we tried those different substitution lines and looked at their longevities, we see no effect of them. So whatever the stress resistance doesn't seem to be explaining differences in longevity for that. So it's unrelated. So another path that has occurred in my lab over the last decade or so is we got involved in this project. It's called the Senile Rhabditis Intervention Testing Program. So the NIH, through the National Institutes of Aging, had created this program called the Intervention Testing Program, where mice are given different compounds and then tested to see if it makes the mice live longer. And so their big hit and their success story that fuels all their future funding is rapamycin. So I wouldn't surprise me if someone in this room is taking rapamycin to try and make themselves live longer. So this is now in the news. It was just in the New York Times, I think, a couple of weeks ago. This is a compound that you can take. It's an immunosuppressant, but it has broad effects, cellular signaling effects. And so that's a hit. But mice are incredibly expensive to work with, and they're slow. And the NIH also wanted to look at more genetic variation as a key indicator of these effects. And so they created this worm project because we can do experiments much more quickly. And so it's a joint project. It's a very interesting kind of logistical thing. So it's my lab, Monica Driscoll's lab, and Gordon Lithgow's lab at the Buck Institute. This is in the Bay Area and at Rutgers. And so we all do the same experiments, not exactly at the same time, but we instantly replicate every experiment. So that's another thing, and I can talk about that. It turns out that aging and longevity is an incredibly noisy phenotype, and it's really hard to replicate these results. So that's one of our important contributions to the field, is just that message. It's like you really need to replicate your experiments. There's a lot of simple-minded storytelling in the aging field, and probably a lot of those results are not true because they're cherry picking. So we've tested a lot of different compounds. About 15, we've tested 77 compounds. 15 of them actually have a 30% increase in lifespan. So that's a pretty big effect. 31 have a 10% effect. I guess by this date, we have 81 compounds tested. So I got to update my brain. So here are some examples of things that we fed worms that make them live longer. Some things like green tea extract is, a nice thing to be drinking on a Oregon winter. Thioflavin T is a protein binding thing. Metformin's another kind of famous anti-aging thing that people are using. It's an anti-diabetic treatment that was discovered, and then some other things that are going on here. I wanted to talk about this one, sulforaphane, because it's kind of a fun story. That's currently our favorite compound to work with for a variety of reasons. I just want to highlight why you do these experiments in worms and not in mice. So my lab is also the center for the data center that coordinates all this stuff. So we have a centralized data repository, and we do all the statistical analysis and all that sort of stuff. So I don't know if you can read these numbers, but this is 700,000 here. So we're adding about 100,000 observations a year in this project. And so it's a lot of data, a lot of experiments that we've been doing. And so, we'll be up to 1 million pretty soon. There's a little plateau here that's called COVID. We couldn't actually do any experiments then, but otherwise it's been a pretty steady rate. So it's a lot of things we're just cranking through. So here's sulforaphane, and this is pretty typical of what our experimental results look like. So sulforaphane is a compound that's derived from broccoli. And so, it's popular. It turns out that as we've gotten into it, people have been studying sulforaphane for a long time for health benefits. But surprisingly, they hadn't really looked at these longevity effects very extensively. So here's a worm lifespan. The black one right here is the control. And then we have various concentrations of sulforaphane. And what you see is that around, I think, 100 micromolar sulforaphane is the best. We can see it here better. So these are the absolute curves, and this is a change in median lifespan relative to the control. So you see we get this kind of classic dose response curve where there's an intermediate level of sulforaphane that's good, and that tends to be around maximized at 100 millimolar for worms. And we've actually since then done 800, and it starts being a little toxic. So it's, again, a classic thing. Another nice thing that my lab did that I don't have time to go into, why we're really hip on this, is they also started asking, when do you add the compound? When does it matter? And it turns out that you only see these positive effects when you add it early in life. Late life intervention does not help the worms. And I think there's some very broad biomedical stories in general in this field that need to be learned as we move forward. Okay, so what we did is we took a number of different days from the 100 micromolar thing, and we did whole worm transcriptomics of the controls and the treated guys so that we could start building this transcriptomic clock and ask what's going on in terms of gene expression and why are the worms living longer? Is there any functional insights that we could get? And so I'm going to show this in a couple of different ways. This is looking at roughly 20,000 genes in this dimensional reduction approach. So this is 1 dimension, 2 dimensions. And then what I'll highlight is that the diamonds are the controls. So you'll see here's day 2, there's day 4, day 8, day 12, day 16 is the control. So there's this little loop. The shape of that has no inherent meaning. It's just a multidimensional kind of mapping of where variation is. But what I wanted to show you is that the sulforaphane guys start off day 2. Sulforaphane is right next to the day 2 controls, day 4. And then as you get to day 8, you'll see that the sulforaphane guys are slowing down, and the day 12 guys are more similar to the day 8. And then the day 16 guys are very similar to the day 12 controls, and so on and so forth. So it seems like what's happening is that the expression patterns of the sulforaphane guys are looking younger by about 4 days, which is about 25% of the worm's lifespan. So it's, and I didn't really highlight it, but the sulforaphane makes the worms live 50% longer. So it's pretty big effect. So what we did is we took every gene in the genome and fit a time-based model so that we could have an expression clock for each gene. And so the black one is the control. These are just 4 genes that we chose. Turns out they're all related to basically a stress response phenotypes. And then what we did is for each one of these, we took expression of the sulforaphane and then just mapped it back to say, what's the age equivalent expression pattern for that gene? And then that gives us this pattern. So this is the actual age of the sulforaphane treatments. And then this is their predicted age if their expression pattern looks like the control. So the dashed line is the 1-to-1. So that's if they were exactly the same. And so we did the control versus the control. So we asked what its pattern was for itself, which is always a good thing to do. And that's what these little gray things are. So you see that the control expression is always predicting its actual age, because it was not surprising because that's what we created the model for. But what's happening in sulforaphane is that exactly as we saw in the kind of anonymous dimensional scaling thing, is that when we fit an actual model, around about 120 super informative genes, that we see this pattern where the worms are really slowing down in their apparent aging. And actually, day 16 is when the worms are starting to really start to age. So it makes sense that it's kind of asymptoting out. So in general, this is now a big deal. These clock things are a huge deal in the aging field, especially epigenetic clocks. But no one's really done it this way in a very consistent expression thing where you're fitting models and trying to actually look at a gene by gene basis. In particular, rather than getting a single value, you actually get a distribution of ages across all the genes, and then you can ask which genes are most predictive and what's the total variation. So we're very excited about this approach, and so we're implementing this for a number of these compounds. And the idea is, especially through the CITP approach, is that we can then have this uniform set of things all done under the same conditions, and then we can start doing things like applying machine learning and other approaches to ask what are the patterns that actually capture the description. There might be different syndromes that lead to long life, depending on what the compounds are. So here are some of the functional things that are either up or down regulated. I won't bore you with this stuff. There's a KIG pathway analysis. We can do Go analysis, all these things. But in general, what we tend to find are these drug metabolism things like the cytochrome P450s and other sorts of, here's another P450 thing. So these are all systems that are involved in a detoxification response. And so I think this is actually a general syndrome for a lot of these compounds that we find that make the worms live longer. They actually think we're trying to kill them by poisoning them. And so we're not really hurting them, but they turn on all these stress responses that make them live longer, okay? So if you're George Bush, some of you might be old enough to remember that guy, George Bush, who had this funny thing about broccoli. He refused. He said, I'm president. I don't have to eat broccoli anymore. It's like, well, so from one thing, we would say, yeah, George, you should be eating your broccoli so that you can live longer. But his actual physical response, which some of us might have, is like, this is yucky. And so that's kind of what the worms are. I think they think it's yucky, but it's what doesn't kill you makes you stronger, I think is a really central feature to many of these aging results. And it turns out that just right here at OSU, Emily Ho is one of the world's experts in sulforaphane function. And so this article just came out in the New York Times, October 14th. How healthy is broccoli? And second paragraph, Emily is quoted. She's at the director of the Linus Pauling Institute. And so we started talking to Emily about what's going on with sulforaphane. So one problem with worms versus mice is that we can never really tell what the worms are taking in. We hope that they're eating it, and some days they respond, they don't. In a mouse, you frequently either put it in their chow, and so you know they're eating it, or you actually inject the drugs directly into them so you can know what the physiological concentration is. We have a really tough time knowing what the physiological concentration in a worm is. And so Emily's lab is helping us do this through mass spec analyses. And this is still early days, and I won't go through this in any detail, but basically, here are control worms that haven't been fed the sulforaphane, and they don't have sulforaphane in them. So praise the Lord. And then here are guys who've been fed sulforaphane, and they do have it. And then there's various metabolic products of sulforaphane. And Emily's expert at knowing what those things do and how long they hang around. And actually, this graph doesn't show the data, I realize just now. But we also treat the bacteria that the worms eat with sulforaphane, and they have a different metabolic profile. So we're always concerned that the things that we're observing are actually the worm's response to the bacteria's response to the compound, which is, biologically interesting, but probably not as medically relevant. So it turns out that the worms are eating the sulforaphane and that we can characterize it. So we're now digging in on this. This was just the very first experiments that we did with Emily's lab on this. So now we're doing this in a big way across multiple strains, multiple species, all sorts of stuff. So it's another really interesting phenotype that we can look at. And because we're working in worms, if we really want to dig into this, we can look at all these different mutant pathways and really dissect out exactly what's going on. And we are interested in it. All right, I'm going to just quickly run through this last little story, which was led by my graduate student, Zach Stevenson, who just graduated last year. He's now a postdoc at the University of Washington. And that is to start moving towards these kind of one-off things to do things much more comprehensively. And so one of the challenges with animals versus, say, bacteria, yeast, or even human cell lines is it's great. We can do genetic transformation now. It feels relatively easy, but it's kind of slow because you have to inject in the case of worms, you have to inject the DNA in, and you can kind of only do it one animal at a time. And that's because there's very strong separation of the somatic tissue from the germline tissue. It's one of the fundamental ideas in animal development. It's called the Weissman barrier. And so you got to get through that. Whereas in a yeast cell, the DNA and the cell itself are one and the same. And so as long as you get into that cell, you know it's going to end up going in the progeny. That's not true for animals. You actually have to get it into the germline and integrate there if they're going to throw progeny with that. And so it's a slow-ish process. It's a miracle that we can do it at all, but it's slow if you want to do hundreds of thousands of transformations. It's a lot of technician time stabbing worms, and it's not really feasible to do. So Zach invented this new approach to solve a problem that I'll talk about in a second, that utilizes something that's worm biologists have used for a long time, but has not been fully exploited in our opinion. And that is when you inject DNA into C. elegans in particular, it creates an extra chromosomal array. So you kind of get a plasmid in an animal for free. And worm biologists have been using this for years to clone genes and to identify things. But they haven't really done a lot with creating structure in these arrays. And so that's what Zach ended up doing. So first thing that we needed to do is, I'll just really buzz through this, is to create this a good way of having a landing pad where a piece of DNA from these arrays could land in a very consistent way. And then once it happened once in integration, it couldn't happen again. So we use CRISPR-based approaches to do that. And basically we call we use a split landing pad where when the DNA inserts in, it completes this hygromycin gene, which leads to resistance. So we can basically screen worms. If they're hygromycin resistant, we know that the transformation has occurred. And if they don't, we know that it hasn't occurred. So we're basically use homology arms. We use CRISPR to cut open the center of this landing pad. And then we can basically integrate in a little segment of DNA. And we can do lots of things with what that DNA can be. Zach's specific application was to put a barcode in there so that we could track the worms, as you'll see in a second. So our basic thing is that a lot of synthetic biology has been done in single cell systems because you can use transformation to do it. In worms, we put these libraries in of many different DNA elements, and then just through natural propagation, the worms will make those elements, and then we use some signal, frequently heat shock, and then we'll do that, and then all of a sudden, randomly those elements will go into this landing pad, and seems to be quite random in the experiments that we've done. So that allows us to create a diversely tagged individuals, and we can use that in a lot of different applications. So we like to say the transformation creates diversity from the outside in. You have the DNA elements that you then push into the cells. And here we make the worms from the inside out. It's a little diversity bomb that we then let go. And then all the different things come raining into different individuals separately. And so that's why Zach called it TARDIS. If you're a Doctor Who fan, if you walk into the TARDIS, it's this massive thing, even though it looks like a little phone booth, a British phone booth. Okay, so Zach wanted to apply this, and this is why he actually invented this thing, to do experimental evolution. This gets back to the history element of things. And so we have this ways that we can create these cultures of worms, many, many millions of worms in a fairly small volume, so we can do very large selection experiments. Zach has figured out how to crank this up. so that we can do in a single experiment, we can have 4 Californians worth of nematodes. Well, the human equivalent of 4 Californians. I'm sure there's many more nematodes in California than that. But we can do very large experiments this way. So what Zach did, he just to do proof of principle, the things that he injected in were different molecular barcodes that were all unique DNA sequences. And then he tagged them into 2 different backgrounds. One was a wild type, one was a mutant that was that's resistant to ivermectin, which is this now infamous horse compound, but it's used for basically to kill nematodes in livestock. I would not recommend it for COVID, but worm biologists have been studying it for many generations because it's really an important product. So then he went through and then did these competition experiments, took out the barcodes, sequenced them, and then we could trace the lineages. And now this brings together the history and the cross-sectional approaches, the dynamic approaches into one. And so when we have no ivermectin in there, you'll see the blue is the wild type and the red is the mutant. The wild type starts taking over. And as we slowly increase the concentration here, is kind of, there's some noise for sure. This is just single replicates. We're showing you as an example. And as it increases in concentration, then you get more and more red, okay? So resistance wins out. That's not a novel observation. However, no one had actually ever done it at this fine a scale. And so we're actually able to create now this very fine scale response curve with high replication and high precision estimates of these selection coefficients, where we can actually estimate the selection coefficient based on the shape of those trajectories. And so yeast people have been doing this now for about 10 years, but we're now excited that we can apply these approaches to animal systems for the first time. Number one, using barcoding, which is a novel contribution. And then we can also, because it's worms, we can scale up our population sizes to be able to do things now, for instance, to look at the distribution of mutational effects, which is what we're gearing up to do now. So I'll just wrap things up and just give you a hint of kind of where we're going now. So Zach's approach, which allows this large-scale transgenesis, has led us to really think about where we want to go in the future. At the same time, for work that I haven't been able to talk about, we've been using a variety since the very beginning of our work in C. elegans, using mutants and other approaches to ask questions about that are fundamental to evolution. So for instance, the role of sex. So we use mutants that can outcross and don't out and they have the self. And so that was some of the earliest experiments we did. And then my graduate student, Katya Kazamatis, created the system using a more modern approach where we use this Degron tagging thing where we can attach to an essential sperm protein. And then when we add the plant hormone auxin, that thing gets pulled into the degradation system, and then it basically turns sperm on and off. So Katya did a great dissertation where she was looking at the effects of sperm competition by basically using the signaling system that she created to allow to do that. So I have a new graduate student who's starting to think about doing this to create a titratable mutation rate approach as well, so that we can look at these fundamental aspects of evolution using a synthetic approach where we manipulate things. But more than that, we're working very hard to expand Zach's approach to, instead of having barcodes substitute in here, to actually have them be CRISPR guides themselves, so they can start targeting genes. So our hope is that in a small set of lines, we can target every gene in the genome systematically, and then we can do mutant screens on things like longevity, which have been very hard to screen for historically. So this is a proof of concept where we put a guide in our landing pad. It's what's called the DUMPy10 mutation, which is a standard screen. And then it's expressed, it attaches to the Cas9 in there, and then it targets the dumpy gene. And lo and behold, we make dumpy worms, and they're different kinds of mutants and stuff. So we know that it works, but it turns out that we're creating all the molecular machinery. We have to have 2 separate CRISPR events, which turns out to be a major pain in the rear. because we have to CRISPR things to bring it in from the array to the landing pad. And then we need to have that thing CRISPR the gene, and it has to happen in the right order or else the whole system breaks. So that's turning out to be, we thought that would be a 6-month project, and it's been a year and a half trying to work that out. And then the final bit, which I won't talk about at all really, is that now I'm like full on, well, if we're going to do these little things, why not just create a complete synthetic biology system within an animal, multicellular animal? It's never been done before. It's a big deal in bacteria, yeast, and human cell lines, or mammalian cell lines in general. So this is where we're really gearing up, is how do we then start creating artificial circuits where we can do things. So there's two aspects of that for the conclusion of this talk. One is that we can then use that to, for instance, study function. So we want to create little molecular tweezers that we can go in and just change the gene expression of every target of these stress response things and see which ones are really important for driving longevity. But I'm also interested in creating synthetic circuits that we can then subject to experimental evolution, so we can understand how circuits evolve and which structures of the circuits. These are long-standing questions in the evolution of genetic systems that we haven't really had a good way of approaching. So I'm excited about that. So basically, overall, this is the way I view our work, and also I think the way a lot of the future in this field is going to go, is that we'll figure out ways of manipulating function using these synthetic circuits, and then we'll test kind of what these things, how they work using these barcoding approaches, so that we can combine the historical context with high precision selection estimates. And then that will lead us to understand what's functionally relevant, which we can then test more directly through molecular biology, which then will allow us to go and create new circuits that are driving that. And hopefully that will be a little flywheel of insight that we can start generating. And then eventually, try and tie these back to the natural ecology of the worms, but we definitely can't talk about that now. So with that, I will wrap up, and I'm happy to take any questions you might have.
So we have time for some questions if anyone. If there are trade-offs associated with the longevity, like in terms of reproductive output or metabolic rates or performance, given that this idea that you accumulate damage and you can either fix that damage or you can reduce how much damage is accruing, then I'm wondering what you're selecting for, I guess, in that.
Yeah, so we've actually done selection experiments now a long time ago where we selected, I think maybe similar to what Mark has done for early reproduction. And we didn't see a big trade-off in longevity for that. I think worms are a little complex because I did go into their much of their biology. So they're normally reproduced as selfing hermaphrodites, but they can outcross also. But it's controversy in the field exactly what those trade-offs might look like. There doesn't seem to be super strong reproductive trade-offs. But in general, I think the reason why these things are conserved approaches, and why, if there's any hope of these things being generalizable across animals, is for the trade-off that you essentially said. Is basically an energy thing. Are you going to invest in making new offspring, or are you going to invest in protecting your soma so that you can make offspring later? And I think worms turn out to be a very good system for that, because they can essentially go into this hibernation mode where they can live for months before they go. So we haven't explored, especially like in these chemical interventions, what the trade-offs might look like, or even how things affect reproduction. We have looked at that in other contexts, and it's not totally clear. That's another use of this barcoding approach, is that it's never quite clear what phenotype you should look at. And those experiments are kind of hard because worms will produce 300 offspring, so it's a lot of counting. But competitive fitness assays are the best way to really look at that in a fine scale way. And we've worked on that for many years, and I've had former students and postdocs work on that, and they're kind of crappy to do, but I think with the barcoding, we'll be able to do them very well.
I'm curious about the CITP, the intervention stuff. When you say that a compound or a chemical is generally effective, what does that look like across strains or even recombinant versus isogenic?
Yeah, so that's a good question. So there's a big trade. Let's just say that the folks at the NIH it's great that they were interested in natural variation, but they still kind of are a little typological. Because when I came out as a quantitative geneticist, it's like, oh, we'll look at hundreds of lines. They're like, no, I think 3 or 4 is good. And so that's kind of this trade-off in investment. We actually started at my behest. We looked at 24 different isolates, and then we chose 3 that are kind of the most extreme from a genetic point of view. And then we look at those, and then we can go back and look at the other ones. One of the things that is quite concerning about the CITP is that C. elegans is quite druggable, so it responds to compounds very well. We look at 2 other species that are also hermaphrodites, C. briggsii and C. tropicalis. They rarely respond to the compounds. And we don't really know why that is. And that's why this is the work we're doing with Emily now. We don't even know if there's species and strain specific uptake differences that are driving it, or if it's actually the downstream effects that are doing it. So with sulforaphane now, we can at least go and say how much sulforaphane is actually in this worm. And so that's going to be useful for separating out those effects. So they turn out to not be so generalizable, but interestingly, when we see something that works in C. elegans, it tends to work in other systems as well. So that's not always true. Like rapamycin does not make worms live longer for whatever reason. But what we haven't done is, and again, we're just starting to do this with the bulk segregant approach, is we really haven't looked at these complex recombinant populations to see, because we do think, and actually I just got a new NIH grant to do this, because we think that we can map these strain by drug interactions using the same kind of thing by trying to look for extreme longevity effects with and without the compound. So that's an obvious approach to try. There's one in the back there. Yeah.
Yeah, so if you, getting sort of a generalized approach, it's chemical compounds, and it may be some sort of stress reaction or something that's triggering things that fall into place for longevity. Do you think that you could get similar reactions just by changing the environment, like stressing them or maybe exercise?
No. Believe it or not, so my colleague Monica Driscoll has several papers on worm exercise. So you can force them to swim. And if you just dump them in liquid, they don't like that. They kind of thrash around. Exercise does help them, it turns out, live longer. And some of the early experiments, actually by my other colleague, Gordon Lithgow, when he was a postdoc in Tom Johnson's lab, is some transient exposure to high temperatures will also help, number one. And there's some indication, although we have not seen it for these compound effects, that the same pathways that help you be, for instance, resistance to temperature stress are also related to longevity. So I think there's this general, and this is where the field is right now, and is kind of trying to find these very generalizable things. I don't think that's the future of aging, quite frankly, because I think it's just too broad. Yeah, you can take metformin and disrupt metabolism in your whole cell, or rapamycin and suppress all these different systems. But those are likely to be highly pleiotropic. I think the future is, and that's why we're really investing in the synthetic biology, is to be much more precise and to find not just things that drive overall stress response, but which are the subcomponents that are really important, and then to design drugs or other kinds of effectors that just tweak that one little part instead of tweaking everything. And I think it's just very early days. I analogize it to cancer biology. So early on in cancer, research, or not research, but treatment, you would take these chemotherapies that would just blow out every cell that was replicating in your body. And it was good, it made people live longer. But that's not where cancer research is now. It's very specific. Can you target something for this cell type or this particular marker? And aging is at the broad chemotherapy stage right now, much even earlier. So I think we're way, way away, which is why, you know, billionaires investing all this money or people self-treating and all that. It seems like it's not a good idea right now. But I've been wrong before. Sometimes the simple-minded works, but not often.
Alicia, you have a question?
I'm gonna ask about your trench buildings data.
Yeah.
See whether or not there's like an enrichment for specific transcription factor binding sites and that whether or not you're activating like the signaling pathways that are
Yeah, we are looking at that specifically. So there's one particular transcription factor. It's called SKIN1 in worms. It's NRF2 in other systems that is involved in these kind of detoxification systems. And there was this project called ModEncode that tried to look for transcription factor binding of all the, not all, but a number of important transcription factors in worms. So we have those data sets. Unfortunately, those experiments have tended to be done in the absence of the environmental elicitor. So they're just kind of constitutive. And it turns out, at least in worms, there's a lot of double usage of everything. And so a lot of those things are also important developmental regulators, and that they have these other effects. And so I think this gets back to your environmental thing, is you need to understand the function in the appropriate environment, and that goes for binding. So I'm guessing we don't actually know all the targets in the right way. So that's another thing that we're trying to move into in my lab, is to use ATAC-seq and these other approaches so that we can do our own annotations, cut and run, to start looking at it in the context of the phenotypes that we're interested in. So what are these things doing in the presence of sulforaphane? In general, I find that these global kinds of things where you rely on annotated data sets don't lead you very far. You really need to dig in and have experiments that you're doing yourself to manipulate it.
Great, thank you. Thank you all for coming. Thanks for a great talk.